Bismillahirrahmanirrahim. Assalamu alaikum, dear students. The topic of today's lecture is the medicinal chemistry of GIT drugs. So, as you all know that the medicinal condition involving the gastrointestinal tract is mainly the peptic ulcer, chemotherapy-induced emesis, diarrhea, and constipation. So we will see one by one what are the treatments and what drugs are used for the treatment of these medical conditions. So first of all, what is the peptic ulcer? The peptic ulcer is the localized erosion in the stomach and duodenum. What agent are generally used for the treatment of the peptic ulcers? Generally the antimicrobial agents, H2 histamine receptor blockers, proton pump inhibitor, prostaglandin, and antacids, and mucosal protective agents. So we will see one by one some of the examples and of these agents, right, and then how they function. So first of all, the antimicrobial agents are used for example, generally the infection caused by the Helicobacter pylori. As you know that this is naturally found in the stomach of many people, right, and it causes generally the inflammation and leading to the membrane erosion. Generally for the treatment of this inflammation is this inflammation can be treated through the antibiotic, for example the erythromycin, amoxicillin, and tetracycline. So generally what are the structures of these antimicrobial agents? Generally, for example, this amoxicillin, it contains the beta-lactam ring drugs for this. So this is the beta-lactam, right? For the second example of antimicrobial agents are the bismuth compounds. For example, this is the bismuth subcitrate, all right, or a bismuth subsalicylate. The third example of the antimicrobial agent used for the inflammation caused by the Helicobacter pylori are the tetracycline, right? Tetracycline generally composed of these anthracene. So this is the basic structure of the anthracene. So we will see one by one. First, the microbial agent is the amoxicillin, right? So generally it is the derivative of the penicillin, which contains this beta lactam ring. So this is the beta lactam ring, right? And the rest of the ring, which includes the sulfur also, which is called the pinam 4th high 1 bicyclo 320 heptane 7-one. So these type of the drugs are generally called the beta lactam antibiotics, and so these it is very close to the ampicillin. Just only there's a hydroxyl substitution at the para position. Para position introduced into the side chain of the phenyl moiety, right? So just you can remember what is the difference between this ampicillin and amoxicillin, right? Just only the difference of the substitution of hydroxyl group. So this is the para phenolic hydroxy group substituted with the beta lactam ring. Second example is the antimicrobial and mucosal protective agents are the bismuth compounds or the bismuth subsalicylate. So you can see this bismuth here. The bismuth complex is with the two citrate molecule. This is one citrate molecule and this one is the second citrate molecule. That's why it is the bismuth subcitrate complex. And then the second one is the bismuth subsalicylate. So this is the salicylate. This is the salicylate and this is the bismuth. So this is the complex of the bismuth with the this salicylate. So this is generally used for the successful eradication of the H. pylori 80 to 90% is possible with the various combination, right? So generally the Bismuth salt do not neutralize this stomach acid, but they do inhibit the pepsin and increase the secretion of mucus. So this increase the secretion of the to form the barrier against the diffusion of acid, which is the hydrochloric acid in the ulcer. Third antimicrobial agent which used for the inflammation caused by the H. pylori is the tetracycline. So tetracycline is actually the reduced form of the naphthalene. So you can see this naphthalene, this aromatic delocalization, but this is the reduced form of the naphthalene, which is the tetracycline. So this is the family of the antibiotic is characterized by highly functionalized, partially reduced naphthalene, which is actually the 4 linear fused 6-membered ring system. So the most important thing in this tetracycline is the enone conjugation system, right? And the stereochemistry at this 4 carbon, which is the C4 alpha dimethyl amino moiety, right? So first the 2 structural functionality which is very important. One is the stereochemistry at C4 carbon, and the second thing is the conjugated phenolic enone system, which is started from the C12 and then to the C10, right? So this one the enol system, right? So start from the here C12 to C10. This is called the conjugated phenolic enone system. So these 2 things are the structural feature of the tetracycline. So here you can see the stereochemistry at C4 carbon. When it is alpha stereochemistry, then this is the active form of the tetracycline. If the stereochemistry is beta of this methyl amino moiety, then it is the inactive form of the tetracycline. This is the mechanism of action of the tetracycline: interfere with the protein synthesis at ribosomal RNA, so leading to the bacteriostasis, which inhibit the growth of the bacteria, not kill the bacteria. So that's how this tetracycline is bind with the sulfur phosphate group of RNA through the different binding mode. For example, this is pi stacking. For example, this part of the molecule binds with the sugar phosphate through the hydrogen bonding. This is the chelation, metal chelation, and then this one is the ionic bonding. So this is how the tetracycline bind with the sugar phosphate group of the RNA and inhibit the function, inhibit at the biosynthesis at protein biosynthesis at ribosomal level. So now we come to the second part, which is the H2 histamine receptor blocker. So there are 3 type of the receptors, histamine receptor. One is H1 receptor, which is found in the smooth muscles. H2 receptor, which is found generally in parietal cell of the stomach. The stimulation of this H2 receptor in the stomach, which lead to increase of the gastric acid secretion. The third type of the H2 the receptor, histamine receptors, are generally found in CNS. So our concern is the H2 receptors and the blockers. So the local hormone histamine, which is generally stimulate the release of the gastric acid by interacting with a specific type of the histamine receptor, which is called the H2 receptor. So histamine antagonists have proven to be the important anti-ulcer agent, although it is largely been suspended by the PPIs, which block the mechanism by which hydrochloric acid is released from the parietal cell. So we will see what is the structural feature of the molecule which have ability to bind with the H2 histamine receptor. So H2 histamine receptor blockers are generally the molecule which blocks the H2 histamine receptor and inactivate the function of the H2 histamine receptor. So generally the molecule which have reported having ability to bind with the H2 histamine receptor and inactivate the function of the receptors are the cimetidine, ranitidine, famotidine, and niacetidine. So in case of the cimetidine, the basic skeleton is very much same as the histamine, which consists of the imidazole ring, and the other drug which is also have the ability to bind with the H2 histamine receptors are burimamide and metamide. Only the difference are the substitution, different substitution. In case of ranitidine, niacetidine, and famotidine, the basic skeleton is this. Only the difference is the aromatic ring, right? So now we come to the chemistry of the histamine, because in order to develop the drug of the which have ability to bind with the H2 histamine receptor and blocks the function, we should know the structure of the chemistry or structure and chemistry of the histamine. All right, so in unionized form, this histamine has a one neutral and two basic nitrogen rings. So you can see these are the three nitrogen rings of the histamine. So histamine is actually the achiral molecule, but it can exists in multiple conformational state, in which the trans conformation is believed to be the preferred for H1 and H2 receptor. So meaning that they have the staggered conformation in case of the ability to bind with the H1 and H2 receptor. So this is the trans conformation of the histamine molecule. So and then this is the different tautomeric form of the histamine. So this is one tautomeric form, and this is the second tautomeric form, in which the only the difference is the placement of the hydrogen, right? So these are some of the molecules which has been synthesized by looking the structure of the histamine with the different substitution, whether these molecules have the ability to bind with the these histamine receptor, and what will be the effect on the H2 histamine receptor. So you can see this 2-methyl histamine, this molecule is H1 selective agonist, and once you would substitute it at different place, right, it has the ability to bind with selectively bind with the H2 histamine receptor. So the basic features of the H2 receptors antihistamines are the molecule have the this basic skeleton is the histamine, and with the different substitution, right? So generally the drug approach in order to bind with the histamine receptor and blocks the function, it starts from the idea starts from the molecule of the histamine, because histamine have ability to bind with the histamine receptor, and of course it stimulates the function. But once this molecule which have ability to bind with the histamine receptors and block the function, it can reduce or the secretion of the gastric acid. So histamine is made up of the imidazole ring. So this is called the imidazole ring, which is consists of the two tautomeric form, one tautomeric form, and this one is the second tautomeric form, right? The pKa of the amino group, right? So the pKa of this amino group is 9.8, which means that the plasma pH of the 7.4. The side chain of the histamine is 99% ionized. With the once it has a pK value 9.80. So now we will see that what are the basic requirements in a molecule which have allow the molecule to bind with different receptor. For example, for the H1 receptor, the essential requirements are the side chain. Well, this one is the side chain which have the positive charged nitrogen and at least it should have the one attached proton, because the quaternary ammonium salt which is which lacked the such of hydrogen were extremely weak in the activity. So this is the basic requirement, that the side chain which have the amino group at least have one attached hydrogen. And the second thing is the heteroatom, which is this one. The heteroatom is flexible, had a flexible chain. So this one heteroatom has a flexible chain above the cation and the cation and the heterocyclic ring. And the third thing requirement is the heterocyclic, the heteroaromatic ring did not have the imidazole ring. So for the molecule which have ability to bind with the H2 histamine, there is not such requirement they have the imidazole ring, but it should contain one nitrogen atom which have the lone pair of electron at ortho to the ortho to the side chain. So this one is the basic requirement, the molecule which have the ability to bind with the H1 receptor. The basic requirement for structure requirement for H2 receptors are generally it contains the heterocyclic heteroaromatic ring, which contain the this amidine unit, which is the this lone pair of electrons CH and then NH. This is the amidine ring, right? And the nitrogen atom of the aromatic ring bound with the hydrogen bonding, right? And the second thing is the terminal alpha amino group, which involve the binding interaction with the both type of ionic and hydrogen bonding. So meaning that the requirement for H2 receptors are it should have the amidine unit, right? And then this nitrogen bind with the receptor through hydrogen bonding. And this one is the terminal alpha amino group, which bind with the receptor via the ionic or the hydrogen bonding. So now we come to the we will see how it binds, what are the different binding site in H1 and H2. You can see this is how the histamine binds with the H1 receptor, right? So you can see only one nitrogen requirement is required for in a molecule to bind with the H1 receptor, while in case of the H2 it should have two nitrogen which bind with the receptor, because it has three pockets to bind the molecule with the receptor, right? So one is the hydrogen bonding, this one nitrogen, one is the second nitrogen bind like this, and then after this is ionic bonding with the receptor. So it have three binding pockets. So like this you can see H1 it is inactivated, so when histamine bind it will switch on. But for example, in case of the H2 histamine blocker, once it bind in and inactivate the receptor, right, in order to stop the function of the histamine receptor. So this is the first this is the molecule which has been synthesized in order to develop the molecule which have the antihistamine act as antihistamine, right? So this is alpha guanylyl histamine. For this, so this imidazole ring, this one is the histamine. They have substituted the alpha guanylyl group at the terminal nitrogen, and they have found that this molecule have antagonist activity, because it has ability to bind with the extra pocket which is in the H2 histamine receptor, right? So different type of the molecule has been synthesized in order to get the potent antihistamine, right? For example, isothiourea, right? So and then this methyl thio groups. So this in thiourea, the positive charge is now restricted to the terminal portion of the chain, and it interacts more strongly with the antagonist binding site. And the antagonist activity increased, but the compound is still the partial agonist, right? So these are some of the substitution that the researcher have done in order to get the potent antihistamine. Two other analogs were also synthesized, in which this gonadine group has been replaced with this methyl thio group or methyl group, right? But upon the modification of this at this site will lead to the partial agonist but poor antagonist activity. But they have found that if, let's say, modified the terminal part of the histamine, will lead to the antagonist activity. So this is how you can see the terminal part of the alpha terminal part of the molecule binds with the receptor through the ionic bonding and hydrogen bonding, right? So strong bonding, right? And then if, let's say, in case of this methyl thio, the binding is weak because it bind only with the one ionic and hydrogen bonding instead of the two binding site in case of the isothiourea. So this is how the molecules is bind with the receptor. So the properties of the thiourea group is very similar to the gonadine group because of because the thiourea group and the gonadine group both are planar and they have the similar size. So and then they can take part in the hydrogen bonding. But the alteration in the biological activity is reasonable be attributed to the difference in the basicity between the two groups. So unlikely the gonadine, the thiourea group is the neutral group, right? So this is due to the C double bond S group, which is the electron withdrawal group, and then it withdraws the electron from the neighbor nitrogen and make known basic and more likely amide nitrogen. So this is the reason that this is a fact that a neutral group could bind to the antagonist site and not to the agonist site. So in order to bind with the antagonist site, the group should be the neutral, not the ionic group, right? So antagonist site involve the hydrogen bonding. That's why it is for the strong hydrogen bonding, molecule should be neutral at the terminal site. So the further chain extension and addition of this N-methyl group leads the bromide, which you have there. So this molecule is highly specific competitive antagonist of the H2 receptor, and it is 100 times more potent than alpha guanylyl histamine. But it is not suitable clinically because it was still too low for the oral administration, right? So so the pK value of the imidazole ring in the histamine is generally the 5.74, meaning it is a weak base, right? And the pK value of imidazole itself is 6.8, and for the bromide is 7.25. This value shows that the imidazole ring are more basic than histamine and more likely to be ionized. So in bromide, the side chain is calculated to be the slightly electron donating group, electron donating group. Therefore, the imidazole ring in the bromide is more likely to ionize than the histamine, where the side chain is electron withdrawal group. So therefore, the binding of the imidazole ring is important for the antagonist as well as the agonist. it is suggested that the pK value closer to the histamine might lead to be better binding. So for this necessary to make the side chain electron withdrawal rather than the electron donating group. So they come to the conclusion that the side chain should have any electron withdrawal group rather than electron donating group in order to make the side chain more susceptible to the hydrogen bonding. So for this one, they have inserted the sulfur group in order to synthesize the thiabromide and metamide, right? So electron withdrawal group. So these sulfur act as a sulfur is a quite good isostery for the methyl unit, right? And both groups have similar van der Waals radii and bond angle. The bromide have significantly lower pK value, which is very near to the histamine and found to have enhanced the antagonist activity because of this electron withdrawal group, right? The metamide is also the 10 times more active than the bromide, and they showed the promising anti-ulcer activity, but it has a side effect of the kidney damage and a condition which result in the reduction of the circulation white blood cell, and which make the patient susceptible to the injection. So further development has been required in order to lacking the side effects. So with keeping this idea, they have developed the cimetidine. The metamide has a side effect associated with the thiourea. So the better to remove this thiourea group. So they have removed this thiourea group with the derivative of the nitrogonidine and cyanogonidine group, right? So this is the cyanogonidine group. So they have found that the cyanogonidine group was more potent analog and was chosen for the studies. So this is how you can see we introduced the scientific the researcher have introduced electron withdrawal group in order to decrease the pKa value or near to the histamine, right, which can have better ability to bind with the histamine receptor. And then the second thing is they have removed the sulfur group because the metamide have the side effect because of the presence of the thiourea group. So they have replaced with the thiourea group with the cyanogonidine group or alpha-gonidine group, and so this part of the molecule have ability to bind with the antagonist part, right? So that lead to the synthesis of the cimetidine. So you can see in the cimetidine it has this imidazole ring, right? So it has the imidazole ring, it has electron withdrawal group which have ability to decrease the pK value of the imidazole ring. And then the third thing is it has the cyanogonidine group, right, which have ability to bind with the receptor through hydrogen bonding. So generally in case of the cimetidine, this is the side chain of the cimetidine which have contained the cyanogonidine group. Generally out of these tautomeric form, immunotautomer and aminotautomer, this number 2 is the most preferred tautomeric form of the gonadine group, because since the cyano group has the strong electron withdrawal group on the neighboring nitrogen compared to the 2 nitrogen which is farther away. So this makes the neighboring nitrogen less basic and therefore less likely to be protonated. So that's why this tautomeric form is the most preferred tautomeric form in case of the cimetidine. Metabolism of the cimetidine. Cimetidine is itself metabolically stable and is secreted in a largely unchanged. The metabolites which is reported in case of cimetidine is the oxidated sulfur oxidase, right? So the oxidation of the sulfur and oxidation of the methyl group, which is on the imidazole ring, and it has been found that cimetidine inhibit the cytochrome P450 oxidase system in the liver. In case of the cimetidine conformation, out of these ZE, EE, and EZ and ZZ conformation, the conformation Z, E, and EZ conformations are the most stable conformation in case of the cimetidine analogs, right? So the ZZ conformation is not favored since the cyno group is forced. Here this cyno group is forced too closely to the alpha methyl group, so that's why it is not the favored conformation. So another example of the H2 histamine receptor blockers is the ranitidine. So they replace the imidazole ring with the furan, right? Imidazole ring with the furan in case of the cimetidine. So it is 10 times more active than the cimetidine. The imidazole ring is same as in the histamine, but the C4 group, which is methyl group, and the 4-atom side chain include the sulfur. The sulfur atom increase the potency compared to the carbon and oxygen and terminal polar basic group in case of the n-cyanogonadine group. So the gonadine substitution with the electron withdrawal group have significantly decreased the basicity compared to the gonadine group. In case of the niacetidine, instead of the imidazole ring, there is a thiazole ring. In case of the famotidine, this is the thiazole ring, and at the this terminal it has the gonadine group. This one is the gonadine group, and this one is the sulfonylamidine group, right? So this famotidine is 30 times more active than the cimetidine. Now we come to the proton pump inhibitor. So the final step in the acid secretion in the parietal cell of the gastric mucosal is mediated by the gastric hydrogen potassium APTase, right? The gastric proton pump inhibitor. So generally the molecule which have ability to act as a proton pump inhibitor, they are the omeprazole, lanoprazole, and related analog. They produce the inhibition of the stimulation of the gastric secretion. Generally these all are belongs to the class of the benzimidazole. So this is, you can see, this is the benzi and this is the imidazole. They are from the benzimidazole. So all compounds are closely structurally related. They contain they contain the this paradyl methyl sulfonyl benzimidazole, right? So this is the pyridine type, pyridyl, and this is the methyl, and this is the sulfonyl part, and this is the benzimidazole. So all structures are resembled to this skeleton. So the omeprazole consists of the 3 parts, right? One part is the substituted pyridine, which is substituted pyridine, which is methoxy group, and then methyl group and two methyl groups, right? And so the substituted benzimidazole, so this is the substituted benzimidazole, and it contains the CH2. It contains the methyl sulfonyl, which is this methyl sulfonyl. This is the methyl sulfonyl chain connecting the two position of these ring system. So these are the structural features of the omeprazole and related analogs. So these are all molecule. You can see it has this one is the pyridine, this is the methyl sulfonyl linker, and this one is the benzimidazole. All molecule, you can see all molecules have the same structural feature, right? So they have these three most important part. These structural features of the proton pump inhibitor. How they act, the mechanism of action of the proton pump inhibitor is start from the protonation of this imidazole and nitrogen, and after that this nitrogen of the pyridine will attack on this double bond, and it will form the spiro this intermediate. So this one is the spiro intermediate. After this the protonation will, after this the protonation will occur, right? And as a result of this sulfonyl will be converted into SOH, right? And then after this nitrogen of the imidazole ring will attack on the sulfur, right? And elimination of the water molecule will take place, and the formation of the pyridinium sulfonamide structure. So now this structure is ready to bind with the proton pump inhibitor through the SS linkage. So that's how this omeprazole binds with the proton pump. So generally the structural activity relationship, generally the electron donating group on the pyridine group, which is, you can see in this case, this is the methoxy group, is maintain the good effect, right? The substituted in the benzimidazole ring are not so important, right? The most important thing is the electron donating group, right? But the substitution with electron withdrawal group, for example nitro group, or the highly electrophilic group, for example this NHCOCH3 group, have will affect on the low anti-secretory effect. So mucosal protective agents, they are the skull part. So they are the composed of these sucrose octa sulfate, right? So this is the sucrose, and this is all are the octate 8 the sulfate ions, right, which is from the complex with the sucrose, right? So sulfate is the complex of sulfuric acid ester of the sucrose and aluminum hydroxide. So generally this sucrose the sulfate aluminum complex bind to the hydrochloric acid of the stomach and act like as acid buffer with the cytoprotective property. So in this molecule, the most important part is the sulfate ion. If the sulfate ion is replaced with the phosphate ion, then it failed to reduce the acid peptic injury. Antacid these are the generally the weak acids, right? So they are commonly the salts of aluminum and magnesium, such as aluminum hydroxide or aluminum oxide hydrate or magnesium hydroxide. Calcium carbonate or react with the HCl to form generally the calcium the carbon dioxide and calcium chloride. And the last one is the second last one is the prostaglandin. They also have the anti-secretory effect. This is somehow the structure of the this prostaglandin, which is E series. It generally have the structure feature is contains the cyclopentane with cyclopentanone with the acyclic chain, the side chain on a ring, right? And then it has the C13, C14, and C15 and C16, right? So these are the it has the different structural features at this site are made to prevent the rapid metabolic conversion into the inactive products. So these molecules also have the anti-secretory effect. Antidiarrheal agents, opioids are generally used as the antidiarrheal agent. They are the structural modification of the 4-phenylpiperidine, right? So this is the piperidine and this is the phenyl ring, right? And then it lead to the discovery of opioid analog. The most common one is the diphenoxylate HCl, which is the atropine sulfate and loperamide, right? So generally in case of the phenoxylate HCl with the atropine sulfate, they are generally this is the diphenoxylate, and the active form is the diphenoxin, which is actually this diphenoxylate is the ester form of the diphenoxin. So diphenoxin is actually the active metabolite of the diphenoxylate and used as an antidiarrheal agent. So this is somehow the drug receptor binding, how the drug binds through ionic bonding, hydrogen bonding, ion dipole interaction. That's how the molecule of the histamine bind with the H2 receptor, right, through different hydrogen bonding, ionic bonding, or hydrogen bonding. So this is how this bind with the, you can see this is ranitidine bind with the H2 receptor membrane through hydrogen bonding, ionic bonding. This is, and the histamine binding and the activation of the H2 receptor. So through the binding mode, this is, so can see how this almost the binding modes are same, but depend upon the molecule how strongly the molecule bind with the receptor. So that's all for the medicinal chemistry of the GIT drugs. So what you have to remember in this lecture is, you can see that what are the main components, structural feature requirement for in the molecule which can bind with the H2 histamine receptor, or what are the structure requirement for the proton pump inhibitor, right? So these are the two important things you have to remember, right? So do not no one asks you about to draw the structure, just you have to know how the molecule can bind with the receptor, number one, and how, and then what are their structural features, right? How if the two molecules have which molecules have the ability to bind with H1 and which molecule cannot bind with the H1, and they can bind with the H2 receptor. With this, thank you very much. Assalamu alaikum warahmatullahi wabarakatuh.